Let's see if you're comfortable with enzyme mechanism. Because if you're not comfortable with enzyme mechanism, next semester you're going to have a wonderful time. Okay? You're going to be like, yeah. Okay. You're not going to be happy. Like I said previously, enzymes can use ways to stabilize the transition state. If we take a look, and what you'll see is that what enzymes do essentially is it's going to do reactions to the substrate such that it makes the substrate become the product. That's what enzymes do. Okay? So there are several strategies to achieve catalysis. We have electrostatic catalysis, and these are non-covalent bond providing complementary interaction with the transition state, what I just mentioned. Okay? So you can have electrostatic interactions with the transition state. These could be hydrogen bonding, charge-charge interaction, polar interaction, whatever. And these things will be used to essentially stabilize the transition state. They're used to stabilize the transition state. And then you actually have this thing called covalent catalysis. And what these things are is that they are covalent bonds formed between the substrate and the enzyme into substrate-enzyme complex. Because as you will see later, a lot of reactions are in a way, pretty complicated with the chemistries that they do. So then in this case, your enzyme actually has to come in, do reaction, actually have chemical interaction with the substrate, and then form the product. Okay, so you will make covalent bond temporarily between the enzyme and the substrate. You will actually form chemical bonds between enzyme and substrate. It's just that at the end of the catalysis, that chemical bond is removed. And you have acid-base catalysis, the movement of protons. It's super important in catalysis mechanism. Why do you guys think we spend so much time on pKa's? This is one of the reasons. You have to understand what kind of protons are easy to be removed from a molecule. If you can understand that, if you can get a feeling for what kind of protons are easily removed, then you can easily assess the likely mechanism for an enzyme. Next, you have proximity and orientation. Okay, so we talked about that previously with the delta S, right? So proximity and orientation works by promoting the molecules into the right position for reaction. Let's say you want to react fatty acid. You want to react fatty acid right here. Then the orientation, of course, matters. Let's say I'm going to use ATP with fatty acid, and I'm going to turn that into fatty acid phosphate, okay, something like that. Now, ATP has to be on this side, not on this side, right? It makes sense. Orientation matters. So enzymes can also increase the rate simply by putting the substrate next to each other. And then we also have distortion of the substrate and the active site. Okay, so that's the induced fit model right there. A good enzyme, a successful enzyme, will use all of these strategies to enhance the reaction rate. Okay, you'll see the example that we're going to talk about, chymotrypsin, will use essentially all of this. Okay, a good enzyme uses all of that. Minimally, okay, what's the minimal that an enzyme has to use? Proximity and orientation. Okay, that's like the minimum. Okay, you can actually show that as long as you can create a shape, okay, that binds to the transition state, and then you don't do any of the above, it still increases the reaction rate. Okay, so simply having the correct proximity and the orientation, you can get very good rate enhancement or catalysis already. But a good enzyme will use all of this stuff. Before we go on and talk about catalysis mechanism, there are a couple of things that I want to make sure that we are, on the same page. First thing first, what you will see a lot in biochemistry, like I'm not even kidding, right? 说真的哦，这很认真哦，很认真跟你们讲。What you will see a lot in biochemistry is reactions with carbonyls. Okay, I think by now in your organic chemistry class you have learned everything you should learn about carbonyls. In biochemistry, you are going to see so much carbonyl chemistry, you're going to be sick. All right, a couple of things. You should always remember carbonyl carbon, this guy, is going to be a electrophile because it's bound to an oxygen with 2 bonds. So if you look at the electron spread, what you'll see is this guy is significantly more partial negative, this guy is more partial positive. Okay, that's just how it is. What you will see a lot is essentially some kind of a nucleophile. Nucleophile typically have lone pair electrons or are negatively charged, and it's going to attack the carbonyl center. It will attack the carbonyl center. And then you will form the transition state. So the transition state of this reaction is essentially the second where this guy, okay, attacks this carbonyl center, and then the electrons are going to go up. Okay? And then this transition state is the thing that has to be stabilized by your enzyme. Okay? So, number of things. Number one, you should be very comfortable with the arrow pushing whatever mechanism. Okay? So arrows represent movement of electron. You always go from electron rich to electron not rich places. Okay? Electron rich, electron poor. Now, so let's say you have an enzyme catalyzed ester cleavage. Okay? Ester cleavage meaning what we're going to do is we're going to remove this OR thing and then turn it into an OH. So what you have is you are going to have some kind of intermediate nucleophile that will attack the carbonyl center such that you end up with this tetrahedral oxyanion intermediate. This guy is not the transition state. This is actually an intermediate inside the enzyme. It's not super stable, but it's stable enough for it to appear. The transition state is the place that is really unstable. So right before the tetrahedral intermediate, this is what you have as a transition state. So transition state is something that you cannot capture. Okay, you cannot capture the transition state. Now, what you have here is I want to make this electron attack this carbonyl center, and let's say this is the transition state. What can I do to stabilize the transition state? So obviously you have a partial negative right here. So you can imagine if you have some kind of a positive right here, that can stabilize your transition state. Okay? So here it says that transition state might be stabilized by electrostatic interaction with the active site amino acids and or metal ions, or it might be stabilized by general acids or base. You'll see that in just a minute. Turns out enzyme catalyzed ester cleavage. So again, this is the transition state, and what you see is that around the oxygen you have things like zinc or amino group that's protonated, and you got a positive charge. So you have very good interaction between the negative and the positive such that you promote the movement of the electron that was here to go up. Another way that you can stabilize the transition state is by using general acid and base catalysis. What does that mean? In a way, it's similar to electrostatic stabilization, but here it's a special type which we call acid stabilization, okay, or acid catalysis. Here you have histidine essentially, and you got a protonated histidine, and this guy is essentially a proton. And it has a positive charge. The proton is fairly positive, so then what you want to do is you want to draw the electrons toward that way. And then simultaneously, the electron donating group right here, this proton is going to be drawn that way. Okay, so what you see, by the way, same thing here, okay? So what you see is that you have an acid, it's negative charged, it wants to take this proton. If I take this proton, then the electrons on the oxygen and the hydrogen, the electron between the O and H, is going to go to the oxygen because oxygen is more electronegative. And then now this guy will have a lot of electrons, and then this electron will go to this partial positive. And then what you see is that this facilitates the movement of your electrons. So we're going to talk about enzyme reactions. Now, there are some amino acid residues that you should be familiar with, okay, or at least you should be comfortable with. These are glutamate aspartate. Why? Because it's an acid. This is the proton donor, so you can donate this guy. Or actually, for the most part, in physiological condition, it's going to be carboxylic or carbosylate. It's going to be the anion form. It will draw protons towards it. You also have lysine arginine. These things are going to be positive. Or, yeah, so this you can be NH3 positive, right? These things are going to be able to pass electrons in such a way that your amino nitrogen has the proton or without the proton. Cysteine is a thiol. The pKa of cysteine is not very high. It's about 9, roughly speaking. So given the accurate surrounding, you can deprotonate the cysteine and you make it into S-. Now, cysteine you will see almost everywhere. You will see a lot of cysteine catalyzed reaction because cysteine, when it's in the general base form, it's a very, very good nucleophile. Okay, so it's going to react with a lot of stuff, especially carbonyl carbon. You also have histidine. We talked about how histidine, the pKa is about 6, something like that, and that's a very good trait because pKa around 6, meaning that proton is very loose. It can on and go off very easily, on, off, right? So histidine is frequently used as a proton sort of a transfer station. You will see in the chymotrypsin example in just a minute. Similar to cysteine, you also have serine. Serine is an alcohol, and the alcohol can be deprotonated, but compared to cysteine, serine is usually harder to deprotonate, but nonetheless, it can be. So serine is also used as the active residue, the active site for many different reactions. Right? So the behavior of cysteine and serine, you have similar behavior. And then lastly, you have tyrosine, which essentially it's the same thing. You can remove that proton and you form an anion here. Right? So these are the guys that are frequently observed in amino acid acid-base catalysis. So again, these are the acid-base catalysis. And why are these guys important? Because the protons here, the hydrogen proton, 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 so all of them can be removed. So that's an important thing right there. Okay, so the protons that can be removed will be used in acid-base catalysis. So in biochemistry, when we say acids and bases, that's essentially what we're talking about. Acids have proton, bases don't have proton, okay? You should expect to see a lot of these amino acid residues in enzyme catalysis. So reaction mechanism, so this is what I was talking about with the earlier slides, all right? So again, whenever we talk about reaction mechanisms, you should be very familiar and very comfortable with the movement of electrons. Given any chemical, I give you whatever chemical there, you should know who is the nucleophile and who is the electrophile. Anything that has a negative charge, probably that's going to be a nucleophile. Why? It has electrons, okay? It has a negative charge. So, things that have negative charges, like what? Negatively charged oxygen. For example, deprotonated alcohol. When serine is removed of a proton, it becomes this. Similarly, you have this negatively charged sulfhydryl group. Thiol, removing a proton, becomes S-. Right, so this is serine, this is cysteine. Sometimes you have a carbanion. Okay, carbanions do not appear that much. You also have something that people always forget. Uncharged amine group. This could be amino group, or it could just be an amino group that's attached to something. So why is this guy a nucleophile? Because it has lone pair electrons. So if you use uncharged amine, react it with something, then this amine this nitrogen will have 4 bonds with 1 positive charge. Imidazole, same thing. Okay, this is the functional group for histidine. Okay, we said that the pKa for this guy is 6. Okay, meaning this nitrogen right here is very easy to take a proton, give off a proton at physiological condition. This lone pair electron can do that too. Lastly, you have this hydroxide ion, okay, just OH. This is deprotonated water. You will see this a lot too. Why? Because there are a lot of enzymes that do what? Hydrolysis. Electrophiles, right? Electrophile, number 1, proton, okay? Proton is like the simplest thing you can ever have. Now, in the water, of course, it's not proton, it's hydronium ion, but whatever, we can just say proton. In addition to protons, what do you have? Right? So in biochemistry, for the most part, we only see 3 types of electrophile. They are carbonyl-carbon. Like I said, you will see a lot of carbonyl-carbons. Almost always, your reaction works by having one of these guys reacting with this. Then, in addition to carbonyl-carbon, you have imine carbon. Imine group, in a way, it's very, very similar to carbonyl. Why? Both nitrogen and oxygen, they are electronegative. Okay, now, in the case of imine, usually the nitrogen has to be protonated. Why? Because like if you compare, this is more electronegative, this is less, so the electrons go that way, okay? Then your electron go here, and then this guy will become partial positive, or very partial positive, and then you can have a nucleophilic attack. Lastly, you'll also see this, phosphate, okay? Phosphate you see a lot in like ATP, ADP, organic phosphate, whatever, okay? Now, the phosphorus at the middle of the phosphate would be an electrophilic something, right? Electrophilic atom. Right? The reason, simple, it has so many oxygen around it. Okay? So the electrons are being pulled away such that this guy will be more partial positive so that you can have a nucleophilic attack to the phosphorus center. For biochemistry or biochemical reaction, these things that you should be familiar with. Some of the acid-base chemistry, you should be familiar with charge-charge interactions. So, throughout our entire class, we've been discussing all sorts of different interactions, right? Here, I'm going to show you a complete catalytic cycle of an enzyme, and the enzyme we're going to discuss is chymotrypsin. Okay, so chymotrypsin is like your best friend. It cuts proteins. Okay. So chymotrypsin, we say that it's a type of serine protease, okay? And serine protease What it means is it's protease, so it degrades proteins, okay, or peptides. And we call it serine protease because it's a protein, or it's an enzyme, right, where the active nucleophile is a serine residue. Protease is a class of enzyme that breaks peptide bonds. Serine protease is called serine because the active nucleophile is going to be a serine. Okay, so nucleophile, right? So it's going to be something that looks like that. It's not cysteine, right? And so it's going to be that. This is a classical example of how enzymes catalyze their reactions. So we're going to discuss the mechanism of chymotrypsin. So this semester, at the beginning, we just went through different characteristics, different properties of different biomolecules, right? So this is the first enzyme mechanism that we see. Next semester in biochemistry 2, you're going to see like 50 different enzyme mechanisms, okay? So try to absorb as much as you can. And what you see in the future is that all of these mechanisms are relatively the same. Couple of things as we're going through the catalysis. I want you guys to pay attention to the stabilizing factors that enzymes provide for the catalysis to occur. Okay, we're going to talk about several things and then pay particular attention to what each of those things do. Okay, so chymotrypsin, it's an enzyme, right? So it's the gray thingy that's the enzyme, and the blue thingy that's your substrate. And then you're going to have catalysis around here, okay? Now, chymotrypsin is a protease, remember, that breaks the peptide bond at the location of a bulky residue, right? So it cuts phenylalanine, tryptophan, leucine, tyrosine, those large molecules, okay? So again, chymotrypsin is going to cut those bulky side chain, and it's going to cut the bulky side chain on the C terminal. It's cutting on the right side. Now, a couple of things. This is what the enzyme is going to look like. And this enzyme has a what we call catalytic triad. So this guy, this guy, this guy. These things are the catalytic triads. Okay, meaning that they are the 3 amino acid residues that are really important in this catalysis. Around these 3 residues, this whole position, this entire thing, we call that the active site. Remember that active site is the location which catalysis occurs, okay, on the enzyme. Okay, so active site. First thing first, chymotrypsin is going to cut only the bulky residues, right? Because if it cuts everything, it's not chymotrypsin. So how does it decide where is the bulky residues, right? Turns out in chymotrypsin, you have this hydrophobic pocket right about here. And this hydrophobic pocket is going to bind very nicely with the bulky hydrophobic side chain of your substrate. Okay, so this is the polypeptide substrate. And let's say this guy is phenylalanine, right? So it's going to come in, bind right there, okay? What's the reason they bind? Hydrophobic interaction, van der Waals, whatever. It fits, okay? This pocket fits your R group, okay? So it fits. Let's start looking at the first thing that's going to be involved in catalysis. Right? So just having the phenylalanine bind, that's it, it binds, right? Now, a couple of things. What you see here is aspartic acid. Okay? Aspartic acid, under physiological condition, it's always negatively charged, right? Because the pKa is like what? pKa for aspartic acid is 4, right? The amino acid is pKa 2, but the aspartate, R group, this is the R group, okay? R group, this one is 4. pKa is 4, whatever. At physiological condition of 7, it's going to be deprotonated, okay? So this guy is deprotonated, right? So if this guy is deprotonated, normally it's going to be a negatively charged. Right next to it, you have a histidine. And remember, histidine is going to be able to grab protons. Now, that is referring to here, right? Right here, you have what we call a low barrier hydrogen bond, because N to H is a polar bond. And then here you have a negative charge. Because they're really close, it's going to sort of try to take the hydrogen so that this bond becomes even more polar. So this low barrier hydrogen bond, we can essentially consider that that hydrogen is sort of shared between the nitrogen and the oxygen. Okay? So you have this negative charge that's grabbing this proton. So the catalytic triad, we have serine, histidine, aspartate. And having the catalytic triad is a general characteristic of proteases. And the identity of the nucleophile, the base, and the acid may vary depending on the different proteases that you're working with. And now, for serine protease, the nucleophile is a serine. And in this case, you have a general base and a general acid. Acid could be aspartate or glutamate. Base, it could be histidine, it could be like lysine, that kind of stuff. Okay? So catalytic triads will appear in different proteases. When you guys are looking at literature or looking at different things, you'll always see, for example, serine 195 or histidine 57, aspartate 102. Just like when we talked about hemoglobin, right, the H whatever or the histidine whatever, this is referring to the location of the serine on the protein. Location or the number. So serine 195, meaning that it's the 195th amino acid in this chain. And what you notice is that you have 195 right next to 57, right next to 102. How does that happen? Because your protein is 3D. Okay, it's 3-dimensional, so they can actually be right next to each other, even though their numbers is very far apart. Just so you remember. Right after my phenylalanine binds, that means that my peptide chain is very close to all of these residues. Right? So your polypeptide substrate enters the active site. Now, what you have is, since you have this negatively charged oxygen, this aspartate, it's going to grab this hydrogen. So hydrogen is going to go towards this direction. Okay, the hydrogen is sort of towards the oxygen. And then as a result, what you have is, the electrons on here can sort of promote, so this guy becomes electron rich, can promote things like, sharing the electron towards this carbon, and then this electron can sort of go to here, and then you can have an electron that grabs the proton from the serine such that temporarily you will get something that looks like that, okay? And then this is a very good nucleophile. And then this nucleophile will attack carbon-neocarbon, which is a good electrophile, and it's going to form a new bond. Because it forms a new bond, then you're going to have 1, 2, 3, 4, 5 bond. Obviously, carbon cannot have 5 bonds. So one of the bonds on CO double bond, the pi bond, is going to pop up to the oxygen. And then you form this tetrahedral intermediate called oxyanion. Oxy stands for oxygen, oxygen anion. Okay, so you have a negative charge right there. What you notice is that your histidine is now protonated. And the reason why you can do this is because histidine, it has a pKa is relatively neutral, so it's going to be affected by the aspartic acid right there. So the aspartic acid is going to sort of cause the histidine to want to grab a proton. And so it's increasing the pKa of that particular nitrogen, or that particular proton right there, this one. Right? And then what you have is the first oxyanion intermediate. So this guy is sort of stable, right? And then, somewhere here, that's the transition state, okay? And then now we're going to say how you stabilize oxyanion, okay? So it turns out, okay, oxyanion is stabilized, or typically stabilized, by an oxyanion hole, right? And this is common on many, many different enzymes, not just proteases, okay? Oxyanion hole is common to many enzymes, right? Because you have to have a, counter-stabilizing feature on the enzyme to stabilize your negative charge, okay? In this case, in the case of chymotrypsin, you have your glycine 193 and serine 195, okay? Their R groups don't do jack, right? So their R groups don't do anything. But turns out their backbone amino proton, so this guy, remember here we have NH, partial positive, partial negative. So you have a bunch of partial positive proton or hydrogens here. That's sort of going to draw the attention of your negative charge. So what do you have here? This is essentially your hydrogen bond interaction, right? You have hydrogen bond interaction right there because NH, this is a hydrogen bond, and that makes that hydrogen fairly partial positive, and then that fairly partial positive hydrogen will be used to stabilize the oxyanion. So during the transition state, right, remember we saw this a while back. So during the transition state, you have to have some kind of a positive charge, or you have some kind of a hydrogen bond right here, right? So as long as you have some kind of hydrogen bond right there, or some kind of a charge right there, it will be used to stabilize the transition state. The transition state is not the oxyanion intermediate, okay? Although 你也没办法去纯化这个 intermediate 啦, okay? Okay, so this is the first oxyanion intermediate. So in a way, it's stable. It's stable, especially with your oxyanion hold right there. And then you're like, okay, your substrate is attached to the enzyme, right? So what do you see here? This is covalent catalysis as well, right? Because it's covalent linked to your enzyme, right? So you have a covalent bond between your substrate and the enzyme, okay? So covalent interaction as well. Next, you have regeneration of the carbonyl, right? So because you have a negative charge right there, and negative charge wants to go somewhere. So you can have the negative charge right there, comes back down and regenerate the CO pi bond, so the CO double bond. And as a result, your carbon is going to have too many bonds. So what do you do is between CN, right here, so you have too many electrons here. So the electrons between the CN is going to go and grab this proton right there. The bond between the NH electron is going to go back to the nitrogen. And then now this guy, you see how it becomes neutral. Now the electrons are back. And then the proton went up on the peptide. Okay, so now you have a cleavage. This is then a free peptide that leaves the enzyme. So you just chopped off half of your enzyme. So the first step is complete. Right? The hydrolysis, the first step is done. So part of the substrate is broken. So this guy can say goodbye. Okay, it leaves. But the other half of the substrate is still attached to the enzyme. Right? So the other half is still attached to the enzyme. So we got to get that one off as well. Right? So how do we do that? So then after the first half of the substrate leaves the enzyme, and then you have water that's going to come in. So this guy is water. And water, of course, is polar. So it's going to interact with this nitrogen. Again, this nitrogen has lone pairs. So this is a very good nucleophile, right? Has a lot of electrons. This guy has a lot of electrons. And water is superpolar. So you're going to have an interaction right there. And then what do you see? Right? Lone pair electrons grabs this hydrogen, grabs this proton, then this electron. Right now, you essentially generate this, right? You essentially generate that. And then that is going to attack something, right? So this is a good nucleophile again. Very good nucleophile. And the nucleophile is going to attack an electrophile. Where do you see an electrophile? Right? Right here. This carbon. So it attacks the electrophile, and then again, you generate oxyanion. Okay? You generate the oxyanion again. Okay? And you use the same glycine 193 and the serine 195 to stabilize the oxyanion transition state. So you stabilize the oxyanion. You now have, again, a tetrahedral intermediate oxyanion right there. It has 4 bonds. Guess what? It's going to come back down again. Now, when it comes back down, you have the choice of leaving this bond or this bond. If you leave this, which it happens if you kick out this water right there, then you end up this. So it happens, but I don't care, right? There's going to be a portion of the substrate that's going to go the other direction. It's going to go this particular bond. And then this bond, the 2 electrons, are going to go and grab this proton. And then the electrons between this NH is going to go onto the nitrogen. Okay, it's going to go onto the nitrogen right there. You regenerate your histidine 57. And because the electron went to the hydrogen right there, this bond is gone. The substrate becomes the product and leaves. And then you essentially, your serine regenerates the hydroxyl group. And then after that guy leaves, you can do this again. Okay, now, what do you see? This repeated nucleophile attack carbonyl, make oxyanion, oxyanion comes back, somebody leaves, okay? One of the bond leaves, okay? If you leave the original bond, it regenerates the substrate, so that's kind of useless. So then by chance, you will leave with the one that you wanted to leave with, and then you proceed with the mechanism, okay? Proceed with the catalysis. Now, in this case, this is a favorable reaction. It's driven by what? Right, so enzyme catalyze a reaction, right? So you can see this mechanism helps with the speed of this reaction. So polypeptide hydrolysis without catalysis, without any catalysis, it's going to take a long, long time because amide bonds are really stable, okay? It's really stable. So using this, it becomes faster, right? But is this reversible? I don't think so, okay? I don't think it's reversible. Why? Because remember, whenever we make proteins, okay, we actually have to use essentially an ATP. But here, you go from one substrate into two products, right? Two products, more stable. You gain entropy, right? Okay, so this one, the direction, you go from one polypeptide to two polypeptide. The direction is determined based on the thermodynamics. And here, you go from one thing to two things. You know, the two things, it's smaller, so it's going to be able to move better. So you gain entropy. Okay, so you gain entropy. So the direction is affected based on thermodynamics. Here, it just makes the reaction faster. I'm going to give you an exam from last year, and then you'll see how one of the questions is really nasty because I'm going to show you a reaction, and then I want you to show me the mechanism. Okay, and then using pretty much the same logics, and I want you to show me what that mechanism would look like. Okay, so again, please be comfortable with carbonyl chemistry. Okay, always remember, carbonyl, right after you react with the carbonyl center, you generate what? Oxyanion. You don't stop at the oxyanion. Okay, oxyanion is not stable. Okay, so oxyanion, that charge is going to come back down, regenerate the carbonyl. When you want to regenerate that carbonyl, you got to kick out one bond. Okay, you can kick out the new bond you formed, or you kick out the existing bond. So very quickly, we're going to go through some stuff about enzymes. In addition to just the protein itself, in addition to just the amino acid residues, a lot of times enzymes will have other stuff in it. And for the most part, enzymes will have cofactors. Okay, cofactors are little molecules that go into the enzyme and help the enzyme to do catalysis. These cofactors are small molecules. They can be metals. Okay, they could be metals like, for example, iron, zinc, nickel maybe, etc. Magnesium, you see that a lot. Manganese, you do see sometimes. So metal ions. You can have coenzymes, okay? And coenzymes are molecules. Coenzymes are small molecules. And for the most part, they're derived from vitamins. Okay? So all of the stuff that you take, you know, vitamin B, 1, 2, 3, 4, 5, 6, 7, those are used for coenzyme. They are used for making some chemical reactions. Now, cofactors, some molecules are going to have tightly bound or sometimes even covalently bound, right? And these cofactors, we have a different name. We're going to call them prosthetic group. Prosthetic arm, 你们知道是什么？就那个如果你手断掉，你装支假的手在上面嘛，那只叫 prosthetic arm。所以 prosthetic group 就是一支，一个东西插在你的 molecule 身上，它不插在你的 protein 身上，它原本不属于你的这个蛋白，是后来插上去的，对，所以叫 prosthetic group。Cofactors, we can split them into metal ions and coenzymes, and for coenzymes, we have prosthetic groups or co-substrates. Okay, sometimes there are going to be cofactors that can be used as substrates, but we'll talk about those later. All right. These are some of the examples of metal cofactors and their functions. These ions are primarily used for either of the two things. These ions are used, number one, to stabilize the substrate. For example, magnesium II+. Essentially, all of the ATP utilizing enzymes will have magnesium II+. Essentially, every enzyme you see about using ATP, it's going to use magnesium as well, almost every enzyme. Metal ions are often used in oxidation, reduction. In general, we call them redox reactions, which stands for reduction and oxidation. Why? That is because some of these metal ions can do oxidation changes. For example, you go from Fe3+ to Fe2+. Okay, so they can pass and take electrons. So here's a table, I think found in your book, that looks at some different metal ions and then the example of enzymes that use them and the functions that is involved in these enzymes. Now, like I said, magnesium is involved in ATP. And then for iron, copper, molybdenum, and what else? Okay, so these guys are primarily used for redox. Zinc is used to bind another cofactor. Okay, so you see a lot of these different metals around. Okay, so which is why whenever you drink mineral water is better than distilled water for humans, for any life, because in the mineral water that you drink, you have all these trace minerals that you get from the nature. Okay, so these are actually quite useful for catalysis. Coenzymes. Okay. Coenzymes, like I said, we break them into co-substrates and prosthetic groups. Co-substrates are coenzymes that are attached to the substrate and essentially is a reactant for a biochemical reaction. Okay? Some examples are acetyl-CoA and NADH. These enzymes we're going to hear a lot in the next semester when we talk about biochemistry 2. NADH, I think you need to know right now, this guy is an electron carrier. So every time you hear things about blah, blah, blah dehydrogenase, it's going to use NADH. Okay? NADH or NAD is a huge molecule. The molecular weight for NAD is about 780 or 760 or something like that. It's a pretty large molecule. Okay? You have prosthetic groups. Prosthetic groups are coenzymes that are part of the enzyme. So they're bound on the enzyme either covalently or through very well associated interactions. For example, very strong electrostatic or van der Waals, that kind of stuff. So prosthetic groups are essentially attached to the enzyme. They don't come off. Okay? So in this table, there are some examples that are examples of coenzymes or cofactors used for different function. Like I said, this guy here, this is NAD. This nicotinamide adenine dinucleotide. NAD, what it does is it carries H-, so it carries electrons. Okay, so for your purpose, I just want you guys to know there are cofactors, and I want you to know what are their functions. Okay, what are they used for? And I want you to know the difference between prosthetic groups and substrate cofactors. Enzymes. There are a lot of different enzymes. Okay, a lot of different enzymes. And then, since there are so many different enzymes, we got to have a very good systematic way to classify them. Okay, to say, okay, this enzyme belongs in one class of enzyme. This other enzyme belongs in another class. Okay, so typically we're going to classify enzymes based on their reaction type. Okay, don't be surprised, we don't have that many different types. Okay, now enzymes, for every reaction that it's going to do, essentially it's going to have a number. Okay, and in this case we just call it EC number. That stands for enzyme commission. EC system. There are some websites you can look up different EC numbers. We're going to go over one of them called Brenda for the biochemists out there. If you guys are going to go into the field of enzymology, or for example, when I do my research, I do renewable chemical production, I need to know what kind of enzyme that are available in nature that I can use. So this is a database that's super, super useful. It has a lot of useful information that you will want. Okay, show you that in just a second. Now, each enzyme has a number, has the EC number, and each EC number has 4 parts. Okay, so essentially it has 4 parts. Okay, so for example, 1.1.1.1. Okay, this is alcohol dehydrogenase. Okay, and then each subclass contains a particular description. Okay. Now, the major class here, the first number, the first identifier, That's the only one that I require you guys to memorize. There are 6 major types of enzymes. They are oxidoreductase. It does the transfer of electrons. You have transferase, which transfer functional groups from one substrate to another. Therefore, transferase. Oh yeah, by the way, blah blah ase is enzyme. Okay, just like blah blah ose is a sugar. And then number 3, you have hydrolase. Okay, hydrolase involves hydrolysis. Okay, like protease is a hydrolase, right? Because you're hydrolyzing something. You transfer water onto a molecule. You have lyases. Lyases is you add something to a double bond. Okay, for example, let's say you can add something that looks like that, and then you turn that into this. That would be a lyase, okay, because this is reversible. Okay, so essentially it's the addition of groups to double bonds, or the formation of a double bond by the removal of some kind of a group. That's a lyase. Okay. Number 5, isomerases. Okay, isomerases are pretty simple. It's the transfer of functional groups inside a molecule. Okay, so you change the molecule. You don't have any other substrate. You just have one substrate. You change it, I don't know, somehow. Lastly, you have ligases. Ligases is the formation of carbon-carbon bond, carbon-sulfur bond, CO-CN bond by the condensation reaction coupled to ATP cleavage. Okay, so things that use ATP as an energy input to build a new bond, those things are lyases. So we're going to discuss each different enzymes, and we have some pictures. You got some pictures in the book too that shows you what each classes are.